Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The intellectual program, initiated at the Sir William Dunn School of Pathology in the late 1930s, represents a pivotal moment in the history of medicine, a deliberate turn towards the systematic investigation of naturally occurring antimicrobial substances. This endeavour, championed by Howard Walter Florey, gained its ultimate direction through Ernst Boris Chain's comprehensive literature survey, which resurrected Alexander Fleming's decade-old observations on the mould Penicillium notatum. The subsequent work of the Oxford team, a collective of brilliant and pragmatic scientists, transformed a laboratory curiosity into the foundation of the antibiotic age. This essay will undertake a profoundly focused evaluation of their landmark 1940 publication, Penicillin as a chemotherapeutic agent, examining, in sequence, the internal claims, methodological underpinnings, and logical architecture of the arguments presented within that foundational text. The analysis will proceed by dissecting the paper's structure, treating each of its conceptual sections as a building block in a masterfully constructed case for penicillin's revolutionary potential. The paper itself, full of sober scientific prose, builds its world-altering conclusions from a foundation of meticulous, stepwise experimentation, a process. This evaluation seeks to illumine Section 1, the initial characterization and production of a usable substance. The paper commences with a section dedicated to the properties of penicillin, a passage that immediately establishes the project's immense practical difficulties and the team's resourceful approach to them. The central claim here is twofold: that a potent antibacterial substance can be extracted from the culture fluid of the Penicillium mold, and that this substance possesses a specific, challenging set of chemical characteristics. The authors forthrightly describe their material as a crude, impure, light brown powder. The brilliance of their argument begins with this candour. They define their subject of study functionally as the active principle responsible for the observed antibacterial effect, whatever its ultimate chemical identity might be. Their evaluation of its properties is therefore a pragmatic guide to its handling. They claim the active substance is an organic acid, a conclusion derived from its behaviour during extraction. The methodology detailed, conceived largely by Norman Heatley, involved adjusting the culture medium to a low potential of hydrogen value, specifically around 2, to facilitate the substance's transfer into an organic solvent like amyl acetate or ether, and then reversing the process with a buffer to move it back into an aqueous solution. This process simultaneously concentrated the active agent and offered strong evidence for its acidic nature. Furthermore, they assert the profound instability of the substance. They claim it is rapidly destroyed by acids, alkalis, and heat, as well as by oxidants and certain heavy metals. This finding, based on observing the loss of bacteriostatic activity after exposure to these conditions, frames the entire research program. It explains why their extraction procedure had to be performed at low temperatures and with great speed. This initial section accomplishes something remarkable. It presents a litany of prohibitive challenges, while simultaneously demonstrating a viable, albeit delicate, pathway to obtaining a usable product. The claim is that despite its fragility and impurity, the active principle can be isolated into little, dry form, a feat that had eluded previous investigators and was essential for any systemic therapeutic. Section 2, quantifying bacteriostatic potency and spectrum. Following the chemical groundwork, the authors proceed to an in-depth analysis of penicillin's bacteriostatic action. This section's primary claim is that their crude preparation exhibits a highly selective and extraordinarily potent inhibitory effect against a specific range of bacteria. Their work here builds directly upon Fleming's initial observations, yet advances them from the qualitative to the quantitative, a move essential for establishing clinical relevance. The methodology employed was the serial dilution technique, a standard laboratory procedure used to determine the minimum, concentration of a substance that prevents visible bacterial growth. The authors meticulously document the results for a variety of pathogenic microbes. They claim powerful inhibition of gram-positive organisms, including various strains of Staphylococci, Streptococci, and Pneumococci, with activity being clearly observable at dilutions as extreme as 1 part in 500,000. Conversely, they assert the substance's ineffectiveness against the gram-negative bacilli of the coliform group, such as Escherichia coli. This selective toxicity is a cornerstone of their argument, suggesting a targeted mechanism of action that would spare many of the body's commensal bacteria. A more subtle and highly significant claim embedded within this section is that the bactericidal action of penicillin is minimal. Its primary effect, they propose, is bacteriostatic, meaning it halts microbial reproduction. This was demonstrated by showing that inhibited bacteria could resume growth if the penicillin was removed or inactivated by the enzyme penicillinase. Another critical assertion is that the inhibitory power of their preparation is largely uninfluenced by the number of bacteria in the initial inoculum, a point of immense clinical significance, suggesting that penicillin could be effective even in the face of overwhelming infection. This part of the paper successfully establishes penicillin as a uniquely potent and specific agent, providing the rigorous in vitro data necessary to justify the risks of in vivo experimentation. Section 3. The crucial question of systemic toxicity. The investigation next moves to what is arguably the most critical juncture of the entire paper: the assessment of penicillin's toxicity. The central claim of this section is that their preparation, despite its impurity, exhibits remarkably low toxicity when administered systemically to laboratory animals. This was the great unknown that had deterred further development for a decade. The authors' findings here represent the true beginning of the antibiotic revolution. Their methodology was direct and empirical. They administered their brown powder to mice and rats via intravenous and subcutaneous routes at concentrations far exceeding those required for bacteriostatic activity. Their reported observations form the core of the claim. The animals displayed a complete absence of toxic reactions. They continued to behave normally, with no apparent distress or physiological disturbance. This finding alone was revolutionary. To bolster this central assertion, they add a further layer of sophisticated analysis concerning the substance's effect on host immune cells. They claim that penicillin, even at high concentrations, has a negligible effect on the activity of leukocytes. They tested this by observing the phagocytic function of white blood cells in vitro in the presence of their preparation. The finding that penicillin did not impede this vital immune function was profoundly important. It suggested that the drug would work in concert with the body's own defenses, assisting the immune system by halting bacterial proliferation, thereby allowing phagocytes to clear the infection more effectively. The argument presented is a powerful one here. Is an agent of immense antibacterial power that appears to be almost completely harmless to the animal host and its primary cellular defense mechanisms. The cautious, understated tone of the authors, describing these momentous results with simple declarative statements, lends their claims an immense weight of credibility and scientific integrity. Section 4. Pharmacokinetics, and the challenge of expression. Having established both potency and safety, the Oxford team turned to the practical matter of how the substance behaves within a living system, addressing its absorption and excretion. The primary claim presented in this section is that penicillin is subject to extremely rapid absorption into the bloodstream, followed by an equally rapid excretion from the body, almost entirely via the kidneys. This set of findings introduced a significant practical hurdle to its clinical use. The methodology for this investigation involved administering penicillin to animals and then taking blood and urine samples at specific time intervals. The biological activity of these samples was then assayed using the same bacteriostatic tests described earlier. Their data supported the claim that after injection, a high concentration of active penicillin appears in the blood very quickly, but that this level diminishes to almost nothing within a few hours. Concurrently, They found that a high percentage, often over 50%, of the administered dose could be recovered in its active form from the animal's urine. These claims carried two profound implications. First, the rapid clearance from the body meant that to maintain a therapeutically effective concentration of the drug in the blood, a regimen of frequent, repeated injections or a continuous infusion would be necessary. The authors state this conclusion directly, confronting the logistical challenge head-on. Second, the recovery of active penicillin from urine, while highlighting the drug's stability within the body's internal environment, also hinted at a bizarre but practical strategy for the future in an era of extreme scarcity. The urine of treated patients could become a source for recycling the precious substance. This section tempers the previous optimism with a dose of harsh clinical reality, yet its candidness further solidifies the paper's scientific authority. It demonstrates that the researchers were thinking like clinicians, anticipating the challenges of translating their laboratory findings into effective medical treatment. Section 5, the definitive therapeutic trials. In mice, the paper's logical progression culminates in the final, dramatic section, the therapeutic tests. In mice, this is the synthesis of all prior findings, the in vivo proof of concept. The overarching claim is unambiguous and powerful. The systemic administration of their penicillin preparation can protect mice against an otherwise certainly lethal infection with virulent bacteria. This was the moment where penicillin transitioned from a promising substance to a potential miracle drug. The experimental design was a model of clarity and control. Groups of mice were infected with standardized, lethal doses of pathogenic bacteria, specifically virulent strains of streptococci, staphylococci, and Clostridium septique. A designated control group in each experiment received the bacteria but no subsequent treatment. The experimental group received the same lethal infection followed by a course of repeated subcutaneous injections of penicillin. The dosage and schedule informed by the pharmacokinetic studies in the previous section. The results, presented in stark tabular form, are the paper's most potent argument. In the streptococcal experiment, for instance, all 25 control mice were dead within 16 hours. Of the 25 mice treated with penicillin, 24 survived. The data for staphylococcal and clostridial infections showed similarly dramatic protective effects. The claim of therapeutic efficacy is supported by this incontrovertible evidence of survival. The authors meticulously detail the total dosage, the timing of injections, and the duration of treatment, demonstrating a thorough and systematic approach. By successfully protecting mice against 3 different and highly dangerous pathogens, they also expanded the claim of penicillin's clinical utility, showing its effectiveness against a range of gram-positive organisms responsible for a vast burden of human disease. The section provides the irrefutable conclusion to the paper's inductive argument, a demonstration of life-saving potential that would galvanize a massive, worldwide effort to produce and purify the drug. Conclusion in its final analysis. The 1940 paper by Florey, Chain, and their colleagues at Oxford is a masterpiece of scientific argumentation, a work whose profound implications are built upon a foundation of cautious, rigorous, and transparently presented evidence. Through a methodical, section-by-section progression, the paper constructs an unassailable case for penicillin as a viable chemotherapeutic agent. It begins by honestly confronting the immense challenges of production and stability, moves to a quantitative confirmation of its potent and selective antibacterial action, then delivers the revolutionary finding of its low systemic toxicity. The work proceeds to delineate the practical pharmacokinetic challenges of its rapid excretion, while simultaneously using that knowledge to design the definitive, life-saving experiments in mice. Each claim, from the substance's acidic nature to its non-interference with leukocytes to its dramatic efficacy in vivo, is supported by direct, repeatable experimental evidence. The genius of the paper resides in its restraint. It avoids hyperbole, acknowledges limitations, and allows the stark clarity of the data to convey the revolutionary message. It provided the complete scientific blueprint, that enabled the subsequent larger-scale production of penicillin and inaugurated the age of antibiotics, fundamentally reshaping the practice of medicine and the human relationship with infectious disease. The work stands as a timeless testament to the power of systematic, collaborative research, demonstrating how a forgotten laboratory observation, when subjected to determined and intelligent investigation, could yield a substance capable of saving millions of lives, all starting with a fragile, impure, brown powder.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.